You don't. It's not because I think negatively of you. Is that this stuff is really hard. And let's see here. All right, so if you look up here, this is the original schedule. So we were supposed to have a quiz on this Wednesday. So I'm delaying it to Monday, October 7th, and then we were supposed to have the exam on Wednesday, October 9th. So now, if you look at the rest of the schedule here, I'm going to scroll down some. October 14th, Monday, October 14th, there's no Bio 220 class because that's Columbus Day. So then this exam is now going to be moved to this day, right here. And that's a normal Monday class schedule. So for you guys, it doesn't matter. For my stats class, it does. And then I'm just going to push everything down. Okay?
Exam 2 is the 15th.
Excuse me?
Exam 2 is the 15th.
October 15th, yes. Gertrude, what? Oh, hi.
Does that mean that... Slow down a little bit, mister. I mean, Dr. Nancy.
What?
Because if you're going to push everything down a day, does that mean that...
You still have quiz 6 on the 23rd.
So don't worry about, just worry about Unit 2, and then we'll get to Unit 3, okay? All right, so Unit 3, 4, and 5, right, we'll get to it. We're gonna, I have some, guys, I know what I'm doing, all right? I promise you it will be a reasonable way to get through this material. Right? This is probably a better way to get through it than moving it up. If you want, I can move it up. It's no big deal for me. I'll just literally go a whole hell of a lot faster than I was planning on. Gianna wants me to. All right? So Jelly, do you have any questions? All right, so don't look at 3, 4, and 5. Just know that those things are going to get adjusted. The one thing I will say is Exam 5 isn't going to get changed. All right, Exam 5, I pushed it all the way to the end of the semester anyway. I can't push it anymore. You have 8 quizzes total in this chapter. No. So for the next 3 units 2, 3, and 4, and well, actually next 4 units, 2, 3, 4, and 5. There's only 4 more quizzes. There's one quiz in each with each unit. So it's a 1 or 2 chapters, a quiz, 1 or 2 chapters, a quiz, and then the exam. Okay. All right, so that's the announcement there. This syllabus is going to get changed slightly. What is this? And if you look here, so this, I'm going to make an announcement here for this quiz right here is the quiz that is going to get changed. And then the exam is going to get changed too. All right? And then again, I'll change it for you. All right. So this is the integumentary system. That's all that we're talking about here. And for the integumentary system, what you should really do is study all the stuff about epithelial tissue, because the questions, whoops,
Oh my.
Goodness gracious.
Okay, I think lights on.
Your light up looks
I'm
kind
trying to
of cool.
do it a little bit lower. The epithelial tissue, all the characteristics of epithelial tissue will come into play here, except for those characteristics associated with simple epithelial tissue. This is, the integumentary system is stratified squamous epithelial, and then there's additional stuff that we'll talk about. Lots of learning objectives here. There's three major things about it, is the major layers of the epithelial, of the integumentary system, cells and proteins of the integumentary system and what their functions are, accessory structures including hair, nails, and glands, and then the physiology of it. The physiology of it being the integumentary system. There's two examples there. We're going to talk about temperature control on the body, which you're already familiar with, and then vitamin D control. Vitamin D is a very important characteristic, and it's part of the reason why we evolved different skin colors, because we as a species evolved to be in different areas of the world, and so as a result we're responding to different environmental stimuli. So in particular the sun. So if you're living in the northern hemisphere or way up by the North Pole, you get different amounts of sun than if you live down by the equator. So if you look at the accessory structures here, hair, nails, and glands, and when I talk about glands here, I'm talking about exocrine glands, not endocrine glands. I don't know if I even mentioned endocrine glands again this semester. These are all part of stratified squamous epithelium, and here are some of the 5 major functions here. Of these 5 major functions here, protection, physical protection is important. Another type of protection is temperature regulation, because that's protecting the body. Another protection of the body is vitamin D production. And then excretion is getting rid of stuff. Excretion is tied to temperature regulation. Excretion means at the cellular level it's exocytosis. It doesn't necessarily have to be exocytosis, but it's getting rid of stuff. And that stuff will come into 3 different categories. There's water, there's salts, and then there's other waste products. But by and large, those excretion will come, is another function here. And then finally, and I'm not sure if it's most important or least important, I'm not sure about this, but sensation. If you close your eyes and you can feel your hands touching the table, you can feel someone touching your ear if you're not aware that they are, right? So that's sensation. And if you look at this here, so in terms of protection, when you're talking about protection of the skin or by the skin, UV light is the thing that is most influential or most detrimental to us on one hand, but it's also most necessary. If you look at UV light, where my pointer is, it protects against UV light, but then vitamin D production here requires UV light. So you need enough UV light to produce something called vitamin D, or help start that production. And if you get too much of it, you need specialized proteins to prevent that. When you're talking about water loss and prevention against microorganisms infecting them, the critical protein is keratin, and you have to understand where that keratin is, when it gets there, and what, et cetera, et cetera. There are also specialized stratified squamous epithelial cells that are sensory receptors. These specialized sensory receptors synapse with sensory neurons. So later on in the semester, we're going to talk about 3 different types of neurons. There's sensory motor and integrating neurons that are part of the central nervous system. And so these sensory receptors are not at all neurons, but they send a signal that then it gets converted into an electrical signal, and they what's called synapse with sensory neurons. As I said, temperature regulation, and we'll talk about the mechanism associated with temperature regulation more detailed.
temperature regulation is talking about negative feedback. Vitamin D production is another thing that's involved with negative feedback. And then excretion is really these small amounts of waste products. One thing that we release a lot of in this process here is water. So on the one hand, our skin tries to prevent water loss, and at the other extreme, we try to enhance water loss when it's appropriate. Right? So how do we enhance? Why do we enhance water loss? Because we want to lose heat along with the water. So there's three major parts of the skin here. There's the epidermis, the dermis, and either the subcutaneous layer or the hypodermis. If you look at the picture up here, there's the epidermis, the dermis, and the hypodermis. Typically, the hypodermis is associated with the adipose tissue that is deep to the to the dermis and to the epidermis. And if you look at this picture right here, this should look familiar to here, especially if you look at this part right here, and then you look at this part right there. I want to point out that these bumps right here, and these bumps right there, there's a bump there, and a bump there, another bump right there. Those bumps are where my pointer is, are these bumps right here. So this is the epidermis as is labeled, and this is the dermis, and this is just part of the dermis.
So those little bumps are like where the hair follicles.
So they may be where the hair follicles are. They may also be not necessarily where the hair follicles are. They don't have to be. And then again, now the hypodermis is where I call it part of the skin. It's directly deep to it. It connects the skin and the integumentary system in general to the deeper layers typically associated with skeletal muscle. But it connects to those deep structures right here. And so without the hypodermis, you can't really get away. Generalized functions of epithelial tissue. Avascular. There is no blood flow to epithelial tissue. Wait a minute. How does it survive that? Well, it has, so epithelial tissue doesn't have blood flow, but connective tissue does. It's innervated. There's lots of innervations there. Those innervated specialized epithelial cells, and I won't worry about necessarily knowing the specialized epithelial cells because we go into those later on in the semester. It's enough to say that those specialized epithelial cells synapse with sensory neurons, and those sensory neurons then send signals. And those signals can either be pain, can be touch, they can be other things. The epidermis is stratified. There's layers or individual strata, and you need to know what those strata are and the order in which they do and what their function is and what's found there, and we'll get into that in a minute. And then there's this clear dividing line between the basement membrane, between the dermis and the epidermis. Deep to the epidermis is a lot of blood flow. Superficial to the, or superficial to the dermis, there's no blood flow. So it's avascular, that first point right here. That clear dividing line tells you where the blood flow is and where the blood flow isn't. If any of you have ever had a paper cut, it hurts, but you don't bleed a lot. If you ever then cut a little bit deeper, then you start bleeding. What does that mean? It means you've just gone deeper than the basement membrane, and now you've broken a blood vessel. Prior to that, when you're superficial to the basement membrane, these specialized epithelial cells that communicate or synapse with sensory neurons, they sense that paper cut then hurt, and that was the information that was sent to you, whatever that is, stop that hurts, don't do it again. That's what your brain is receiving, and there's very little, if any, blood. It's important to also recognize that there's going to be histology sections, and in this case there's 3 versions of the skin here. There's 3 different versions of it. If you start up in the upper left-hand portion of the skin, you need to recognize that this is stratified squamous epithelium, and in particular the stratified squamous epithelium is this, where my pointer is running, roughly. There's these little ridges, and then every once in a while there's this deep ridge. And if you blow up this area of this histology slide, and then you bring it down here, you can see now there's individual dark areas right here. And what are those individual dark areas?
Are those the nucleuses?
So those are the nuclei, and then if we blow up this area right here, I think it is, we can see individual nuclei, and as those individual nuclei, this one right here, this nucleus, and that nucleus might look cuboidal, it perhaps would look columnar, and if you say that it's either cuboidal or columnar, you're absolutely wrong, because this is the basement membrane of the stratified squamous epithelial, and you do not look at the structure up there. If you look superficially up at the top there, right where it says lumen, right now this is the apical. This is the apical layer right here. Now these cells are a combination. I will soon tell you that they're dead and they're flat. These are squamous, or these are squamous cells. So it's important to recognize where I'm going here, and you're going to see lots of histology sections. I mentioned the different cells. I just was talking about the stratified squamous epithelial, the cuboidal nature of them at the basement membrane. As the cells get pushed up and up further away from that basement membrane, they start to die, and they change in chemical structure. So there's there's a couple major cells, but there's two in particular here. There's keratinocytes and melanocytes. Keratinocytes is the cell that is responsible for the waterproofing protein keratin. So keratinocyte is the thing that produces that protein keratin, and then it exports it, and this is typical of melanocytes as well. They export the keratin, import it into other cells, and then slowly but surely that keratin gets built up into the cell, and that cell then dies. Melanocytes are the things that, one of the things that helps contribute to skin color. We'll talk about in a little bit. Those melanocytes then will transfer melanin to the adjacent cells, and then those cells become darker, and then they change in chemistry too. There's other cells as well. There's also part of the integumentary system is fighting infection or protection, and so that protection is a physical barrier, and then there's also immune cells here called Langerhans cells. I won't spend a lot of time asking about Langerhans cells nor Merkel cells, because those are things that are for other parts of the class. Later on this semester, we'll definitely talk about Merkel cells. So as these cells move outward more superficially, they get filled with keratin, and then as they get filled with keratin, that's called keratinization. And as they get filled with keratin, they move superficially and they eventually die. That forms a layer that is impermeable. That impermeable layer here protects from abrasion, and as you protect from abrasion, you're also losing cells. So if you have bacteria and viruses that land on your skin, and you also have ectoparasites. How many people have eyebrows? Good. I'm glad that everyone's raising their hand. Right? Including me, everyone in this room, has these specialized eyebrow mites, these ectoparasites. They go like this, they drill into the base of the follicle, and they eat your skin. And then they go to next door neighbors and they have babies, and so you have whole bunches of ectoparasites that live on your face. Right? How many people are grossed out by that? Right? So everybody has it, and it's just part of what our body is. What do they eat? They eat these cells that have died, and sometimes they will, as their cells that are dying, they'll eat that, and they'll eat other material too. As they're eating those, then those cells that get sloughed off, then they might get lost to the environment as well. Bacteria and viruses, other invading particles will also get lost to the environment. And so as a result, as your skin sheds those cells, you're basically protecting yourself. So let's talk about the structure of the anatomy of it. There's 5 layers of the epidermis. There's a deep layer and there's a superficial layer. There's this name, the deepest layer here is the stratum basale. I won't use this term, stratum germinativum. There's the stratum basale, and going deep to superficial is the stratum spinosum, granulosum, lucidum, and corneum. So yes, you need to know the order. These are not all created equally. They're all not all the same. Some of these are more important than others, and we'll talk about those right now. But for right now, this is just part of the most superficial portion of your skin. Okay, now, it doesn't mean that all of your skin is superior. I said superficial, not superior. Where in your skin, think about anatomical terminology, anatomical position, where in your skin would the superficial skin be inferior? What the hell is he talking about? Where on your body would the superficial skin be inferior?
It'd be like fingertips.
The fingertips, yeah, that's one place. The bottom of your feet. In all cases here, we're talking about superficial, closer to the surface here. So the stratum basale is deep. It's the one that is furthest away from the surface. The stratum corneum, where my pointer is always adjacent to the lumen of the skin. The lumen is something that I talked about when we're talking about epithelial tissue, and that's always that's a function here of it. So if you look at these, now we're going to talk about these in order. Some of these layers will have a lot of detail. For instance, stratum basale will have a lot of information, others will not. So which ones are most important? Which ones have the most information? The stratum basale is a single cuboidal layer of cells. It can also be columnar, it doesn't really matter, because it's not squamous. So we don't even worry about what the shape is. We do worry about what it does. It's got very high mitotic activity, and so that means that this is a classic mitosis and cytokinesis. So when you see this in the context of the stratum basale, you're talking about mitosis and cytokinesis producing new cells, and the way this works is that the stratum basale is rooted to the basement membrane, and so the stratum basale, if this is the basement membrane and I'm a stratum basale cell, I go through mitosis and cytokinesis and I push out the other daughter cell that way, and then I go through the cell cycle here. I go again through mitosis and I push it out again, and slowly but surely I'm pushing out the cells out the door. How quickly do I do it? More or less every 19 days. It doesn't really matter. Right? I'm not going to worry about how quickly. Right? The one thing I will say is that the stratified squamous epithelial is very, very mitotically active, especially in context to other cells, in particular nervous cells, cardiac muscle, and then other muscle cells too. Another thing, so these daughter cells are pushed to the surface here, and as those daughter cells are pushed to the surface, they become keratinized by keratinocytes. The other thing that all of these cells have is desmosomes and another series of cellular junctions, and those cell junctions are characteristic of epithelial tissue. They physically hold those cells together adjacently. So the cells that are adjacent to one another, they physically hold them together. And then while they're alive, they allow the cells to communicate. So gap junctions will allow the cells to pass glucose from one cell to another, and if they're both alive. If they're not alive, then it doesn't matter, right? But the desmosomes and the tight junction will remain. Desmosomes and tight junctions are physical connections here. As the stratum basale pushes those cells in between 40 and 56 days, somewhere in that range, so in a month or two, those cells then are sloughed off. They're lost to the environment. If any of you have dust in your rooms, dust in your house, dust in my house, a large portion of that is the cells that you produce each and every day. So the next most superficial layer is the stratum spinosum, or the spiny layer here. There's not very many layers of it. It's between 8 and 10 layers of these cells that are beginning to be flattened. These will contain desmosomes and can be lipid filled. And what does lipids do? Lipids are hydrophobic, and if they're hydrophobic, that repels water. And so it repels water in one direction towards to maintain the water, to keep the water in, but it also repels the water to keep the water out from superficially. Right, so that's important. And then keratin continues to develop in these cells. Next layer up is the stratum granulosum. It's not a very deep layer, or a thick layer rather. There are these preliminary keratin containing proteins here, these granules here, and as these stratum granulosum move further and further away from the stratum basale, they begin to degenerate and die. As the cell dies, right, it goes to the next layer, the stratum lucidum. The stratum lucidum is found in a certain couple of areas here, and when it's found in those areas, that's called thick skin. If it's found in other areas, that's called thin skin. So if you're in a thin skin area of the body, which is a huge portion of the body, we're going to say that the stratum lucidum is either very limited or absent. Okay, so it's limited or absent in thin skin. It's found in palms and soles, and generally in thick skin. And then finally, the most superficial layer is the stratum corneum. That stratum corneum has keratin here, and then there's keratin fibers. They're in the process of being removed from the body or lost to the body, and as that is being lost, they take other stuff with it that they don't want, that the body doesn't want. So that's the general flow of cells from deep to superficial. You should be able to identify individual layers and tell me where they are in the structure of the epidermis. You should be able to order those from deep to superficial and superficial to deep. And you should be able to tell me what is happening in each of these layers, or sublayers, perhaps. You should also be able to tell me where they are, whether it's in a drawing in the middle here, or a histological section here. Now, if you look at this histological section here, where my pointer is a really, really, really important point. Everything above this pointer, that way, is superficial. Everything that way is deep. Now, this is not a nice even, this is a nice even layer right here. This is not how our body works. We don't have a nice even yellow layer between the two epidermis and dermis. So these bumps right here are something that we'll talk about in a minute. Right? Those bumps mean something, and they help with cellular processes. So you should be able to tell me where the epidermis begins and where it ends. It ends up at the top and down here. This is where my finger is in the dermis. Where my red pointer is in the epidermis, and in particular, it's either at the basement membrane or the stratum basale.
Is there a reason why the epidermis is more pink?
So, I mean, superficially?
Yeah.
So, in terms of the way that you make these slides here is you take a little bit of tissue and you have to stain it. And so, what those stains will do is they'll attach to keratin, and then it'll turn that keratin pink or that red. So that's
that was the actual tissue color.
yeah, no. So this is an artificial color. And so if we were to look at an actual skin cell right here, so I'm going to cut a piece of Jaharlan's hand off, right? And then we're going to look at it under the microscope. I don't know, I might get in trouble if I do that, right? But if we were to do that, so don't worry, I won't do that. We wouldn't be able to see nuclei really clearly because they're not stained. And so the nuclei are clear. There are not a lot of obvious, so we would be able to see one nuclei once in a while, but nothing like this. The other thing to remember is that this is epithelial tissue, and this area right here, I'm going to tell you in a minute, is not epithelial tissue anymore. And if it's not epithelial tissue, then the structure is different, and the characteristics are different. And I'm going to tell you in a minute that this area right here is connective tissue largely. And that connective tissue, as a result, has a whole bunch, all the different cells of connective tissue, all the different characteristics of connective tissue, all the different fibers of connective tissue begin to be present in this area right here. And if you look at the picture in the center or on the left-hand side of the slide, this is sort of the characteristic drawings of these cells. These are the first two layers here, the granulosum lucidum, the narrow area right here, and then the stratum corneum. So the histological section this is a little bit more of this. So on the left-hand side, where this period right here is an area, and then if we look at this, let me see, I think it's, if we blow up this area right here, where my pointer is right now is not in epithelial tissue, whoopsie, right? Not in epithelial tissue. Right now it's in epithelial tissue. This is the basement membrane and the stratum basale. I would never say to you, is this the stratum basale or the stratum granule, right? I wouldn't say specifically, like, I'm not gonna put it on the border and say, tell me where it is. Unless this border right here, this border between here and here, definitely easy. Now, if you look over here, what are these cells right here? This is going to be the stratum corneum. So for histological sections, I'm not going to ask you where it is specifically, right? I'm going to be really, really easy about that. For drawings, this is very easy for me to ask. This is not a class in interpreting histological sections. So if you look at this picture right here, so there's this color right here. Now this is purple. Why are they different? Different stains. So there's thin versus thick skin here. Once again, we say that the thin skin doesn't really have any stratum lucidum. Because it doesn't have very much of any stratum lucidum, it's more flexible, and that makes sense. The thick skin is part of the stratum lucidum. It is particularly adept at areas where there's lots of friction. And one of the things that happens if there's a lot of friction here, so it says here, there is the thick skin is got the stratum lucidum present, and I said earlier there's like 2 to 5 layers of stratum lucidum, but there are also areas if there's a lot of friction here. Back in the old days when I was about your age, I used to work at a lumber yard, and I would be loading trucks and doing grunt work, picking up stuff, cleaning things. My hands were battered and beaten. And so I had lots of calluses. And if you have a lot of friction in an area here, that area is going to develop a callus in response to all that friction. The alternative is not to develop the callus and then to get the skin to basically make cuts, right? And your body is trying to respond to that additional friction. That additional friction is going to, if your body doesn't respond, then you're going to start losing lots of blood and you're going to get infections and stuff like that. Again, that's the function of the integumentary system, to prevent that from happening. One of the things I like to say is that I'm one of the, so on the one hand, every one of us have the same amount of melanocytes in our bodies, but one of us is melanin challenged.
Oh.
Guess who that is?
Excuse me.
Well, I didn't say, I'm not pointing anyone else out, I'm just pointing me.
Okay, but I'm right here too.
Oh, well, so I will tell you that one time, my first speech ever that I did in my life was at URI as I think a freshman, and I was taking Spanish 104, and I had to do a speech in Spanish, right? So now I know no one else in this room speaks Spanish, and I was reading about Copa Mundial 1982, and Italy was the winner of this World Cup, I think it was. Anyway, I get up there. Oh, there's a podium. I get up to the podium, and I'm nervous. I'm nervous as hell. So what are you going to do when you're nervous as hell? Your heart's going to start beating. You're going to start sweating right here. And it's a 3-minute speech, so it's not very long, right? And actually, this story is probably going to take longer than my speech. Right? So now I'm like this, and I got it all on index cards. I don't have to have any pictures or anything. I even knew all the guys in Italy, all the winners, the team members of Italy. I could tell you their names and stuff. And I'm starting to shake. And I'm starting to shake I hear something, you hear that little ding, 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 ding? Well, the whole podium was lifted off the table, and I was like, oh shit, what's that? Oh, it's the podium. All right, now I gotta push down, right? Well, now I'm pushing down on the podium, I'm sweating my ass off, and now I gotta go like this, and I have to switch my card because I'm at the end of the card. So I go like this, and I'm like this, and oh shit, I don't wanna lose the cards because then I'm gonna look like an idiot. And by the way, Margaret was in the back, right? I got no riz. Right now, right? I got no riz now. I had even less then. I was sitting next to Margaret back then, and I really wanted to have a date with her, right? So now I'm trying to show off. Well, right? And I get through it. I don't throw the cards all over the place. I survive. In that 3 minutes, I can feel sweat dripping off my earlobes. I'm like this. I didn't have glasses. Hey, yeah, I caught it. I didn't have glasses, right? I'm like this, right? I sit down next to Margaret. She goes, wow, that was awesome.
Is Margaret your wife right now?
No.
Oh.
I don't remember what her last name is. I remember she was a tall blonde-haired woman, or a girl at that point. And I never had any date with her. I was friends with her, and that was it, right? Anyway, the point is that my skin color changed. I didn't change the melanin. I didn't change the melanocytes. I didn't change, there's another protein here that some of us lighter skinned people it's more obvious to, is keratin. But I did change something else in my body that was obvious. It happens to everyone in this room if you're stressed out, if you're embarrassed, if you're hot. Right?
You get red.
Right. Everyone, well, wait a minute. Everyone in this room, it happens to everyone, whether you can see observably whether you get red or not is another issue, right? Is what's the amount of melanin in your skin. So there's pigments here. There's a couple of sort of the presence of pigment is defined by either melanin or keratin. And if you have a lot of melanin, then you're darker skin. If you have less melanin, then you're lighter skin. If you have no melanin, then that's called albinism. And that's a genetic condition. In all cases here, whether you have melanin, a lot of melanin, or very little keratin, or very little of it, all of that is basically designed, it's basically evolved to try to respond to the amount of UV light in the world. So melanin itself is a protein. It's produced by melanocytes, and those melanocytes then, I'll talk about the process in a minute, but those melanocytes make that melanin, and they insert it into the keratinocytes. And then as the keratin is getting developed, it then changes the color of the cell. Melanin is this protein that has this amino acid called tyrosine. So now if I start talking about amino acids, you should know that amino acids, the building block of amino acids, or proteins are amino acids. They're not carbohydrates or nucleic acids or anything else, right? So the amino acid tyrosine is responsible for a major function of melanin, right here. The melanocytes then package this stuff in the cell. The melanocyte is a cell, as that site name means, and it makes that package called a melanosome, and it, through exocytosis from the melanocyte, and then endocytosis into the keratinocyte. And where did we learn all about that? Chapter 3. It begins to change the color of those keratinocytes. Depending on the particular genetics of a person, right, they can either be more or less dark. They can be a little bit reddish or a little bit lighter, right? And there's a couple of different types of proteins. For right now, we're really just worried about melanin, and keratin is another thing. Keratin definitely got a genetic component, but keratin has also got a component of diet too. So let's worry about melanin here. First of all, melanin is rooted in the adjacent to the basement layer, so it's sort of near the stratum basale. It's going to go through this normal protein synthesis here through the rough endoplasmic reticulum. That protein synthesis, that protein then is going to get transferred to the Golgi bodies, and through, it's going to get packaged as a vesicle and through, called a melanosome, and then through exocytosis, that's going to get released. So what did I just say? That's a melanosome right here, and those melanosomes then get picked up through endocytosis into the keratinocytes. As those keratinocytes get more and more melanin, now once it gets into the keratinocytes, now we're just going to call it melanin, and it's no longer melanosome. Once it gets there, then it starts to darken that. So as those melanosomes move into those cells, the cells themselves get darker and darker. And so how do keratinocytes acquire melanin or melanosomes? Through this process right here. How do melanocytes lose melanin? Through this process right here. So there's a number of different ways or reasons for skin color, and it's got just a lot of things here. But one thing that I will say is that the number of melanocytes is pretty consistent between individuals. It's just a function of what do they do with what they're given. I described another area of the, another thing that affects skin color, and that's the presence of blood closer to the superficial portion of the body, not in the epidermis, but if you have red blood cells closer to the skin and you have light skin, then you appear to be red. If you have red blood cells or more blood flow closer to the skin, to the epidermis in particular, and you're darker skin, it's not as obvious. I once had someone say, "Oh, that doesn't happen to me." I was like, "It absolutely happens to you. You can't see it." I said, "I didn't say that, right? You happen to be darker than me. It does happen to you." And I can tell you that if I put you on a treadmill for a half an hour, right, you're going to be sweating. Your skin is going to have more blood flow to there, right? Yeah? So you're definitely going to have that. Right? There's no question about it. So these pigments here can also relay other things, so blushing, anger, inflammation, embarrassment. Me, think about me speaking Spanish, right? It can also appear the opposite for newborns. One of the really characteristic tests of newborns here is a new baby, I remember they did this to my son, they grabbed his feet, and we had a tough birth with him. My wife was in labor for 44 hours, and then she started spiking a fever. And yeah, that's the time when she grabbed my hand, and I thought she was gonna break it, and I said something, and she was like, ugh. Anyway, so it ended up being a C-section because she spiked her fever, and they were really worried about his overall health. And they grabbed his feet, and they pushed down on his feet, and you can see whether or not the color of his feet changed color. And if it was blue, that's bad. Cyanosis is a cause of low or indication of low oxygen in the appendages. If there's low oxygen in the appendages, there's likely also low oxygen in the brain, and that's a major problem. Good thing is that he didn't have that. Although I did have to go to the NICU, the newborn intensive care unit, to see him at like 3:30 in the morning. I walk in and I was like, Mr. Clancy? I was like, yeah, how do you know? Oh, we know, we have your son, he's right here. And there's all these little babies. I was like, holy shit, where did... Anyway, I was dumbfounded, and I was like, oh, hi, bud, how are you doing? And what do you say to a little kid, right? I don't know. He was in this incubator for like a half an hour. I couldn't touch him right like that. And then he stabilized, and they brought him down to mom, and everything was fine. For those of us who have skin, notice I didn't say dark skin. For those of us who have skin, we have the propensity to, potential for developing skin cancer. And there's different types of skin cancer there. If you look at these three different types of skin cancer: basal cell carcinoma, squamous cell carcinoma, melanoma. Melanoma is what? It's going to be, you don't even need to have me tell you, right? Melanoma is going to be a problem, is genetic change in melanocytes. Squamous cell carcinoma is going to be genetic change in those cells that are now flattened here. And then basal cell carcinoma are going to be the ones that the genetic change or cancer developing in the basal cells. So which one would you, if you need to have one, right, you have to have one? This one is probably is the best, right? But my mom had melanoma a long time ago, and she survived it, but it was a very, very significant issue here. So basal cell carcinoma is very common. Again, now basal cell carcinoma is the stratum basale. That's misspelled here. It should be basale. There should be an E at the end. And then there's also squamous cell carcinoma. So now we're going to go deeper from there. Talk about the dermis is deep to the epidermis. That prefix epi means something, and in this case, superficial to the dermis. The dermis has all the different, I say that it's largely connective tissue, but associated with connective tissue are the other 3 types of tissues as well. So that's nervous, muscular, and epithelial. So where are the nervous, muscular, and epithelial tissue in this slide? Well, first of all, for the purpose of this slide, where my pointer is running, it's going left to right, and everywhere that I'm not pointing here is epithelial tissue. So superficial to this is epithelial tissue. So this yellow stuff is nervous tissue. Nervous tissue. These fibers right here are going to be connective tissue, and those fibers are going to be developed by either fibroblasts or fibrocytes, depending on where they are. There's, in addition to, in these layers right here, there's also, so there's nervous tissue, there's connective tissue, there's also going to be blood flow there, and that blood flow is going to have a combination of connective tissue and epithelial tissue surrounding the surface of the blood cell. And then there's also going to be muscles that are attaching to the base of these hair follicles, and those muscles are smooth muscle tissue. So there's all different types of tissues here that, again, is deep to this area right here, which is the epidermis. The dermis has 2 major layers. There's a superficial layer and the deep. That's portion of the dermis here, the papillary layer and the reticular dermis. I'm not going to put a pointer right here and say to you which is which. I might put a pointer way, way, way down here. I might put a pointer way, way, way up here and say which part of the dermis is this, are we in? The papillary area of the dermis is directly adjacent to the epidermis. Always, 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 and never is it. The other thing I'll say to you is the papillary layer is much, much smaller than the reticular layer. It's about 75 to 85% found of the dermis is the reticular layer. So the papillary layer has these lots of elastic fibers. It's superficial and it's not very strong. So if there's elastic fibers that's superficial and not very strong, it's basically in helping respond to different pulling in different areas right here, and so it helps respond to their or return to initial position. There's a whole series of touch receptors here. So there's a whole bunch of different nerve endings. There's a whole bunch of different nervous tissue in there. So there's an awful lot there. And again, I'm not going to ask you for individual ones there. The reticular layer has elastic fibers, but it also has significant amounts of collagen. Collagen is that most abundant protein in our body. In addition here, there's also, you will find areas of that look at first glance like the sweat glands or hair follicles here in that general area, but in fact, that's part of the epidermis, and we'll talk about that in a little bit. Another thing that's present in the skin in general, and the epidermis and the dermis in particular, are these cleavage lines. Generally speaking, here, if you as a surgeon, and if any of you are going to operate on me, if you operate on me, parallel to the incision, parallel to these tension lines, so that it's easier to repair itself, and then you just have less infection and stuff like that. So you want to generally do the operation, if you can, parallel to these tension lines. Those tension lines are where the pulling is going to be happening, so that they're going to be pulling parallel to the lines and not. And if you do it perpendicular, like I have up here, that is going to be more likely to stretch. And if it's more likely to stretch, that's going to be more likely to break. So those collagen fibers are oriented in a particular way generally. And and we have all of those different cells that we mentioned already in part of the dermis. Right? So now we'll talk about the hypodermis. So the hypodermis, the most obvious thing present, cells that are present there are adipocytes, which have adipose tissue or lipids that are found in there. And we talked about adipose tissue. Now that's one type of connective tissue. There's also fibroblasts and macrophages, which are other types of cells of connective tissue that are found in the hypodermis. If you're looking at this picture right here, where my pointer is, largely is where the hypodermis is. And by the way, if you look in this picture right here where the dermis is, we have muscular tissue right here. That's another of the tissue that's found there. And then there's also lots of blood vessels. There's lots of adipose tissue deep to the dermis, so it's about half of the body's adipose tissue here. Three major functions of adipose tissue here. The one that's most important is presence of energy storage or energy source. And when we're talking about energy here, now we're going into the mitochondria and producing ATP. Also insulation, reduction of heat, right, or reduction of heat loss, and also physical protection. So this area of this, if you look at where my pointer is, right here, this area of this adipose tissue is going to prevent heat from getting superficial there. And then all of this blood flow, if you increase the amount of blood flow to the superficial area right here, then that will increase the amount of blood flow or heat to the superficial area. So this will try to prevent it, and then under appropriate conditions here, additional blood flow will increase the temperature loss. The one thing that's characteristic of the hypodermis is both the collagen and the elastic fibers are going to be running in many different directions here. So they're not going to be running, there's not going to be simple cleavage lines like you saw in the previous slide. And this is just a bigger picture of it. You should be able to identify all of these things here. And now what we're going to talk about in a minute is accessory structures of the skin, one of which is the hair, and there's also glands here. Before I do that, I want to talk about what are the terms here. I want to talk about terminology associated with medical terminology here. So what is intradermal versus subcutaneous versus intramuscular? Which one's deep, which one's superficial? Right? Now, if you look at the picture on the right-hand side, this injection is deepest because that musculature is deep to the hypodermis, right here. And so that's a hypodermic needle, or that's a hypodermic puncture right here. That's an intradermal musculature, so you have to go to a really steep angle. Why shouldn't we have an intraepidural injection?
Intraepidural?
Yeah. So there's an intradermal, right? So there's an intramuscular, there's a subcutaneous, there's an intradermal.
Well,
Why shouldn't we put an intraepidural
Wouldn't
injection?
the injection just leave the body, like the skin?
Yeah.
Because it's
Why?
very superficial.
It's so thin.
So it's,
They die off and
what's the majority of the cells doing in the epidermis?
Oh,
They're already
push.
dead.
Shedding.
Pushing.
They're already dead and they're losing, right? So that's the reason why. Also, physically, it's really hard because the skin's pretty thin, right? So there's two reasons there. Of these, which one is most familiar? Probably the subcutaneous injection, right? Intramuscular, there's other injections too, right? So those names mean something, and they're all in relation to the skin. So now, for the rest of this,
lecture, we're gonna talk about other stuff associated with the skin, but for right now, I wanna take a break. It is, let me see here. Whoopsie,
I just recorded myself speaking to my wife for 74 minutes.
No, wait.
Should I send it to her?
Give her the lecture.
Should I send it? I should send it to her. I can tell her all about Margaret.
Oh my god!
So,
That's bad.
well, what's the big deal?
He's already married. What's the big?
I'm married. I've been married for I don't know, 25 years now.
kids,
So I don't know.
Yeah, but you don't wanna make
I
her a
couldn't pick
widow.
Margaret out of my lineup right now.
Who's Margaret?
That girl that I wanted to have a date with when I was a freshman in college. Big deal.
College.
Yeah, 9:43. So how about 10 minutes, 9:55? Come back.
Okay.